All right, hello, and welcome to the lecture on membrane transport. So for this lecture, we're going to be looking at how transport happens across a cell membrane and what mechanisms we have that are going to allow for substances to move across. So transport from inside and outside of the cell is very important. This is going to give us the ability to take in ions, nutrients, everything that we need to have cellular processes exist. So for membrane transport, we're going to use transport mechanisms that are either going to be passive or active. A passive mechanism is one that takes no energy. It is going to be a spontaneous process. This can be either diffusion or osmosis. Diffusion is a movement of solute, usually down its concentration gradient. Osmosis is the movement of water, and that's usually going to be down its concentration gradient. Diffusion happens by either simple or facilitated means. Simple diffusion is where we have transport that does not require anything additional. No structural changes, no channels, no pores. We just have simple diffusion of substances leaking through the membrane. Facilitated diffusion is that which requires an additional transporter. So this is going to be an additional channel, a protein, some sort of a binding agent, a receptor that's going to flip and transport a molecule through the cell membrane. Now that facilitated diffusion, again, we can have a channel that is going to open and be specific for one target, and we're going to have a carrier that is going to be specific for one target. If it's an active process, this means that it requires energy to move forward. So it's not going to happen spontaneously. You have to have some form of cellular energy that's used in order to transport something against a gradient, whether that be a mechanical gradient or a chemical gradient. For active transport, we have primary and secondary. Primary active transport is utilizing our cellular energy source, which is adenosine triphosphate. It's going to allow for a substance to move across a channel against a gradient or against a concentration barrier. Secondary active transport is going to be utilizing that energy from a primary source to piggyback onto that and allow for a secondary molecule to move through. So the energy that's used from the primary transport mechanism is utilized by another source to get across a barrier onto the other side. Now that can either be a symporter, which is going to be similar or the same, meaning moving in the same direction, or an antiporter, anti meaning opposite or against. So that's going to be two substances moving in opposite directions. For an active transport mechanism of vesicles, this is going to be vesicular transport. Vesicular transport can either be going into or out of the cell. If we are going out of the cell, we are simply going to fuse that vesicle with the cell membrane. It is going to open up and release particles into the extracellular environment. And this is known as exocytosis. If we are going to bring material into the cell, we have three different ways we can do that. All three of them are termed as endocytosis, which endo, inside, bringing material into the cell. We can have phagocytosis, which is going to be cellular eating. This is going to be creating a pseudopodia or fake extensions or appendages around material in the extracellular environment to bring it in and engulf it into the cell. We have penocytosis, which is creating an invagination in the cell membrane. It's going to drink in or pull in fluid from the extracellular space and create a vesicle around that space. And finally, receptor mediated, where we have a receptor protein or some sort of signaling agent on the surface of the cell that's going to be responsive to a particular molecule or substance. And once it comes into contact with that, it will form a vesicle around that substance. So this is a chart that we will go through each component of and fill in by the end of the lecture. So for each transport mechanism, we want to characterize and also show what the details are. So for passive transport, this is our example of diffusion. Initially, we have a large concentration of solute, which is dissolved particle, in this extracellular space. Now we can see in the cytoplasm or the intracellular fluid, there's very little. Over time, if this is able to pass through the lipid bilayer easily, there will be an equilibrium established. So for small particles like oxygen, carbon dioxide, this easily happens, where we have diffusion across the cell membrane without the use of any sort of a channel or pore, and we can find equilibrium pretty quickly. So over time, we have diffusion of solute into the intracellular space until we reach an equilibrium. Now at equilibrium, we don't have any net movement of molecules, which means that there's no net change across the barrier. That does not mean that there's no movement at all. It simply means that there's no change in concentration. There is still exchange of solute happening across the barrier, but it is going to be at a 1-to-1 ratio. So it's going to maintain an average. So here's our example of carbon dioxide and oxygen. In the lungs, we have simple diffusion that occurs. The oxygen from the atmospheric environment is breathed in. And in the lungs, we have exchange of that oxygen with the blood of the capillaries. The oxygen diffuses across the cell membrane easily and goes into the capillary space, whereas the carbon dioxide that is brought to the lungs by the blood vessels now diffuses out through the cell membrane out into the lung space where it can be breathed off. So oxygen brought in easily diffuses across. There's a higher concentration of oxygen in the atmospheric space that is brought into the lungs. There is a higher concentration of carbon dioxide in the blood, as that is the waste product from our metabolic processes. So we have an easy diffusion process moving from high to low concentrations and in the opposite direction. There is no energy needed here. There is no transporter. It is just simply movement of molecules across the membrane. For facilitated diffusion, here we have to have something that's going to allow movement through the bilayer. So for instance, we have a sodium and potassium leak channel here. These ions are usually going to be in large concentrations for sodium outside of the cell, for potassium inside of the cell. There are leak channels, which are consistently open, that are going to be specific for these ions. These leak channels are proteins, and these proteins are specifically designed to allow for a conformational binding and movement of sodium and potassium, respectively. A leaky channel is one that is always open, so there's always a small amount of movement going in the opposite directions here. If we find what we term as a gated channel, for instance, here we have a glucose transporter. A glucose transporter is going to be one that is going to be inhibited when there is no glucose present. When there is glucose present, we have usually, in the case of skeletal muscle, insulin that creates sensitization of the transporter. Transporter will open, it will conformationally change and be able to bind to glucose molecules in the extracellular environment. Once it does, it will pull those glucose molecules through the channel and will open into the intracellular space. So you can see with glucose not present, the top of the channel is closed. If glucose is present, we have opening of the top of the channel and movement of molecules through. Osmosis is the movement of water via diffusion. So water molecules are always going to be a solvent. It's what the solutes are dissolved into. Water molecules have their own net movement, however, and this is osmosis. Water is going to move down its concentration gradient. Water moving down its concentration gradient, absent of any other outside influence, is going to be a simple diffusion mechanism. So if we have simple diffusion, we can see that we simply move across the cell membrane, the phospholipid bilayer. We move that oxygen and 2 hydrogens of the water molecule through into the intracellular space if there's a high concentration outside of the cell. So we don't need any additional mechanism here. We just have simple movement. We can also have water movement through a channel-mediated process, and this is by aquaporins. Aquaporins are inserted into places like the distal tubules of the kidney, in response to hormonal innervation. So if we have this insertion of aquaporin channels, now we have diffusion across the membrane utilizing this aquaporin. It'll be able to bind to the water molecules and transport them back through in a place that normally would not be available for movement of water across. Now, movement of water is going to be tightly interrelated to what we term as tonicity. Tonicity is what a solution does as far as its solute concentration to change volume and pressure. So for instance, in this first example, we have the same solute concentration on the inside and the outside of the cell. Now imagine that the solutes are dissolved in water, which they are in all biological circumstances. So those solutes dissolved in water are existing in an equal concentration. This is termed an isotonic solution because we have no net movement of solute in or out of the cell. We have movement, we have exchange, but it's at a 1-to-1 ratio. If we switch that, we change it to having, let's say, a low solute concentration outside of the cell. Well, if we have an equilibrium that's no longer established, the water that's inside of the cell is going to start to move. The water that's inside of the cell is going to move to the outside of the cell. Now, in a hypertonic solution, if we have a higher solute concentration outside of the cell, the water that's inside of the cell is going to start to move to the outside of the cell. So for a hypotonic solution, this is going to cause the cell to swell up. For a hypertonic solution, this is going to cause the cell to shrink. Let's look at that in real time. If we're using the example of a red blood cell or an erythrocyte, during a circumstance of isotonic solution, we have the same concentration of solute in the extracellular space as we do inside of the cell. This means that there is no net movement, so we maintain the original shape and size of the erythrocyte. There's no net movement. That does not mean that there is no exchange. So still remember, there is movement, there is exchange, but the net movement remains zero. For the hypotonic solution, now we have a lot of solutes inside of the cell versus not as many in the interstitial fluid. Well, what does that mean? That means that the solute concentration inside of the cell needs to be diluted. And in order to reach an equilibrium, the water that's outside of the cell is now going to flow into the red blood cell to try and reach an equilibrium of solute concentration related to the outside environment. This will cause the cell to swell up. Now, the opposite of that, if we have a hypertonic solution, there is more concentration outside than inside. So in order to try and reach an equilibrium, the water inside of the cell is going to leave. Once it leaves to reach equilibrium on the outside, the cell itself is going to lose fluid. It's going to lose volume and lose mass, and it will begin to shrink. If we have a red blood cell that's going to swell up, if it does not stop or if there is no equilibrium reached over a period of time, it will continue to swell until it bursts. And if it bursts, this is termed hemolysis. So in this microscopic image, we are seeing a couple of erythrocytes that are reaching or nearing this point of bursting or hemolysis. If we have the shrinking mechanism occurring during a hypertonic state, this is going to be known as crenation. Crenation is the process of shrinking or shriveling of a cell. And if it shrivels down enough, it will cease to be working properly and die. Now, active transport is going to mean movement across either a mechanical or chemical gradient against what would naturally be occurring. So we have to actively switch the direction of movement, and we have to actively maintain the gradient of movement. So for example here, if we put energy into a system in the form of ATP, we are going to be able to move a solute from low concentration to high concentration. This is the opposite of what would be happening during natural diffusion. So we're pushing against a process that is not favored. So here's our example. For primary active transport, this example is showing a calcium pump. This calcium pump is moving calcium from an area of low concentration inside of the cell to higher concentration outside in the interstitial space. To do this, we are utilizing ATP, again, adenosine triphosphate. This is our main form of cellular energy. That ATP is phosphorylated, which means that the third phosphate is cleaved off. And once it is cleaved off, it releases energy. That phosphate bond, broken, releases enough energy to where we can have a conformational change. And once that conformational change happens on the channel, we can bind to and move calcium to the outside of the cell. Once this movement has happened, the channel reestablishes its original positioning until another ATP comes along and gets dissociated to allow it to change once again. So here's the most important mechanism of active transport that we have in our body. This is the sodium-potassium pump, which establishes the resting membrane potential that we see in most cells. So the resting membrane potential is going to be established because we have a concentration of ions that is different across the membrane barrier. The outside of the cell versus the inside is going to have a different ion concentration. Ions are charged particles, so therefore there is a net difference in charge across the barrier. So the sodium-potassium pump is going to actively move these two against their concentration gradients. There is a high concentration of sodium outside of the cell and a high concentration of potassium inside of the cell. But the sodium-potassium pump is going to move these in the opposite direction. So we're moving 2 potassiums for every 3 sodiums. The initial process is having an ATP bind. That ATP binding allows for opening of a conformational change on the inside of the cell. It's going to bind to 3 sodium atoms. Once those sodiums bind, this is going to allow for opening of the channel. So once we have binding, that ATP has been cleaved, so we had the phosphorylation happen. Phosphorylation is going to release energy. The energy is going to allow for the sodiums to move through the channel into the outside environment, in the extracellular space. Now, once that channel opens, the outside channel has 2 slots here. And the 2 slots are now available to bind to potassium ions. So the movement of sodium first opens the channel to now allow for potassium to bind. Once potassium binds, it will then go along through the channel as it conformationally changes back to its original state. So the 2 potassium ions are now bound, and they are going to be pushed through, all right? And once they are pushed through, they move into the intracellular space. And now we have our sodium-potassium pump that is back to its original conformation, ready to bind to 3 sodiums again. So this is establishing a gradient that is the opposite of the naturally occurring high to low concentration movement. So this is important because it establishes the resting membrane potential. And this potential is going to keep an electrical charge across the barrier to allow for things like neurological conduction and nerve impulses to go through a cell and activate process of different things, whether that be muscular contraction, whether that be nerve signaling, whether that be glandular activity. So the electrochemical gradient here is established because this pump is moving the ions in the different directions. So we have a charge across the membrane barrier that we measure in millivoltage. The concentration of sodium is much higher outside of the cell than the concentration of potassium is inside of the cell. That means that sodium is going to dictate the resting membrane potential. Its charge is stronger outside of the cell, So therefore, we have a net negative millivoltage across the membrane. Cell resting membrane potential can be anywhere from -70 to -90 millivolts, depending on where we are in the body. An example of secondary active transport. Here we see the example for a symporter. Remember, a symporter is two molecules moving in the same direction. So this symporter is opening and allowing sodium and glucose to flow through. Initially, this is sodium moving through, all right? So we see sodium actively transported. Once this happens, we have opening of this channel. And this movement allows for these two substances to go in the same direction. So the sodium ions can pass through, and once they pass through, the channel can open and allow binding of glucose, and then it will be transported through. If we're moving in the opposite direction, we have an antiporter. In this case, we have a hydrogen ion and sodium ion antiporter. The sodium is going to move through from the extracellular space into the cytoplasm with the cytosol. The hydrogen ion concentration is going to move out. So the hydrogen ion concentration, we have ionic movement out of the cell, and that is against its concentration gradient. So there's a higher concentration of hydrogen ion outside versus inside. Even so, we actively pump the hydrogen outside of the cell against its concentration gradient and on the opposite direction of the sodium that's coming through the channel. So here we have the chart that I initially posted for these processes. Now it is all filled in. So for our simple diffusion mechanism, these are going to be small nonpolar substances. Our examples are biological molecules. Gases are usually easy. Oxygen, carbon, hydrogen, things like that. They're going to move down the concentration gradient. So it is a simple process. It is naturally occurring. It does not need any energy. A biological example, we saw the oxygen carbon dioxide diffusion. All right. It does not need any additional help. It does not need any additional mechanisms. For a facilitated transport, if it is a channel, it can be either a leak channel or a gated channel. If it's a leak channel, it's consistently open. If it's gated, it will need to bind to a certain molecule. It's going to be moving down the concentration gradient. It is passive. There is no energy needed. And a good example of this is a sodium leak channel that we see on the cell membrane surface. Next is a carrier. So channels and carriers are similar. Carriers are also going to move small polar molecules. Good example of this is a glucose transporter, which we see as a GLUT transporter, a glucose-linked transporter. It's going to move down the concentration gradient. It is not energy needing, so it's passive. And it's going to be based on proteins. So proteins are going to be the carrier here. Our last passive process is osmosis. Osmosis is simple diffusion of water. So this is movement of water down the concentration gradients, and it can be either through simple diffusion or an aquaporin, which is a channel-mediated complex. Primary transport, we can have active transport that is either primary or secondary. Primary, we're going to see polar molecules moved. Our biological example is the sodium-potassium pump. Here we need energy. Why? Because we're moving against a gradient. So we're going against the naturally occurring process. So we're going to need cellular energy in the form of ATP to move that process against it. It is an active form. Secondary active transport is going to be the opposite of this. It's going to be the first way, right? So primary active transport is going to have a second molecule or second compound that moves because of the first one or first initial change. We can have two versions of this, a symporter or antiporter. Symporter, two molecules moving in the same direction. Antiporter, one molecule moving in the opposite direction of the other. We are moving against a concentration gradient again, so we have to have energy. Primary process has ATP happen. The secondary process picks up or piggybacks onto the primary process and allows for movement. Our examples. Our biological example for a symporter was the glucose channel. All right, so this is a glucose symporter where we both move glucose and sodium across the membrane in the same direction. The antiporter example was that of the hydrogen sodium bond. So this is the hydrogen ion antiporter. It's going to move hydrogen ions out of the cell while sodium moves and diffuses in the opposite direction. So when we see the cell membrane, we term the cell membrane as a fluid mosaic. And the fluid mosaic model is something that was proposed over 100 years ago as what the potential structure of the cell membrane looked like. It was first described about 1915, and not until about 1970 did we have a good representation of this. Hugh Dobson and James Danieli described it as a railroad track, which was somewhat accurate in 1935, but we really didn't have characterization of it as a whole until about 1970, when we had Singer and Nicholson, who created a meshwork of what the cell membrane looked like as a whole under a microscopic observation. So we can see that it has a lot of different components, which gives it its mosaic quality. It is a fluid mosaic because it is fluid, not necessarily because it is liquid, but because it is able to move and malleate and is easily adaptable to its environment. So we have several different components here. We've got the phospholipids, which create the bilayer. We have interweaving molecules of cholesterol, which are going to increase the fluidity of the membrane. We have proteins, whether they be peripheral, as membrane proteins, as cellular receptors, as extracellular signalers. We have integral proteins, which are going to span the length of the bilayer. Those can be channels for our movement of substances. We have glycoproteins, which are going to be sugar residues attached to the surface of a protein, usually for cellular signaling and cellular identification. And we can also have sugars attached to a fat. So we can have glycolipids, which we have the lipid of the phospholipid bilayer attached to the sugar residue and represented on the cell membrane surface. So our main structure of phospholipid bilayer is the phospholipid. The phospholipid is composed of a phosphate head group linked to a glycerol that has two fatty acid tails. One of the fatty acid tails is saturated, meaning that all of the carbons are single bonded. The other is an unsaturated fatty acid, meaning we have one double bond or a kink in the chain of the fatty acid tail. This allows for it to be fluid as well, because unsaturated fatty acids are more fluid. We have a hydrophilic head group, meaning that we have a head group that likes water. It's going to be attracted towards water. We have a tail group of lipids that are hydrophobic, meaning that they don't like water and they try to avoid it. So this makes sense with our phospholipid bilayer structure, because we have the phospholipid head facing the extracellular or intracellular environment, while the tails are faced inward towards each other, trying to get into the middle of that bilayer, which is inherently dry. So we have the tails actively avoiding fluid while the heads are out facing towards it. Plasma membrane size is very, very, very small. We would expect that to be the case because it's only a small part of the cell. So usual plasma membranes are only 5 to 10 nanometers in size. If we want to compare that to an actual cell that we've seen previously, we can see that red blood cells are approximately 8 micrometers. 8 micrometers is almost 1,000 times wider than our 5 to 10 nanometers that we had for the plasma membrane thickness. So the membrane is going to be very small comparatively to a whole cell. Now our phospholipid qualities, again, as I described earlier, it's going to form a bilayer. Why? Because those tails are trying to avoid water at all costs. So if it forms a bilayer around a fluid or liquid substance, then it creates a cell where we have components in the intracellular environment and we have an extracellular space, and these are separated by the membrane. If we do not have an internal cavity, we simply have a sphere of phospholipids. They will float around and form a ball. They will form a ball with their heads facing outward towards the fluid and the tails all facing inward towards the central point, trying to maintain their dryness. That hydrophilic head group is going to point towards the inside or outside aqueous environment. The hydrophobic tail is going to face inward towards the middle of the bilayer. And this is going to inherently give us a two-layer cell membrane structure. It will automatically form this structure without any external input because of the properties of the head and the tail. So our main components of the phospholipid bilayer: the cell membrane made up of phospholipids, interwoven with integral cholesterols in between the phospholipids. We have integral proteins. Integral proteins are going to be embedded through the phospholipid bilayer. Many of them form channels or carriers. We have peripheral proteins, which can either be peripheral on the inside or on the outside. These can serve as receptors. These can serve as intracellular messengers. They can serve as external communication components, things of that nature. And we have carbohydrates. The carbohydrates are going to be linked to something. So they're going to be linked to a protein or a lipid. In the case of a carbohydrate linked to a protein, it's a glycoprotein. In the case of it linked to a lipid, it is a glycolipid. So when we see simple diffusion, the concepts that influence diffusion are going to be the same that constitute or influence most other physiological processes. Our first thing is going to be how quickly it's going to move. So the quickness of something moving to equilibrium is going to be dictated by a lot of different things. It's going to be dictated by temperature, pressure, size of molecule, charge of molecule, opening of the membrane, diffusibility, permeability of the membrane, things like that. So first off, we have to determine the steepness of the gradient. If there is 10 times the amount of stuff on the outside of the cell than the inside, it's going to initially move faster than something that only has 5 times the amount of stuff on the outside versus the inside. So if we have a very steep gradient, we're going to move very quickly at first. Once we get towards that equilibrium, the closer we get to it, the closer we get to slowing down the process. So as we reach equilibrium, the rate of diffusion becomes less and less and less until we finally get to the middle. Now, another concept is going to be the size. The mass of the molecule that is moving is going to dictate its speed. Heavier molecules are going to be slower, so they will reach equilibrium with more time. Two other concepts we talked about. Temperature. Diffusion rates are usually going to move faster at higher temperatures. Metabolic processes are quickened or heightened by temperature increases. Same thing goes for extracellular to intracellular movement across the membrane. Particles start to move faster at higher temperatures. Diffusion rates will increase. Now, density. Density and size are not exactly synonymous. If we think about the density of something, if something is very dense, the diffusion rate is going to decrease. If something is very light or non-dense, it's usually going to be faster. So, for example, if we take a solute of sodium, and we say that that weighs 10 whatever units put there. So, oh, you got a 10-pound sodium solute. If we take the same size molecule, say we've got calcium, but we say that the calcium now weighs 2 pounds. Well, their size is the same. They have the same circumference. They look the same from the outside. But the sodium is very dense. It's 5 times denser than the calcium. So what does that mean? Well, that means that the sodium is going to move at a slower rate because it has a higher density. So it might be the same size, it might be the same circumference, it might be the same height, width, whatever, but its mass is going to be much more. So the density increases in density, decrease rate of diffusion. Now, diffusion is going to be the main mechanism that we utilize to get things in and out of the cell. So we have to be able to do this in order to keep ourselves alive. An example of diffusion gone wrong is going to be in the case of dehydration. In the case of dehydration, you are in a hypertonic environment. You're in a hypertonic environment because in the extracellular space, the fluid is now very, very concentrated. In order to stop that concentration, the fluid from inside of the cells is going to leave, and it is going to go to the extracellular space. This will cause crenation of those cells. So the cells will begin to shrink and shrivel. If this goes unchecked, then eventually the cells will shrivel up and die. So over time, we will see that severe dehydration leads to several symptomologies. Usually neurological impulses are the first to go. Dehydration will lead to unconsciousness eventually, and then possibly coma if we have diffusion rates that are very, very slow. So to recap, first is facilitated diffusion. Remember, that is either going to be a protein channel or a carrier that is going to move something that is specifically targeted across the cell membrane, and it's going to be specific for that substance. If it's a carrier, it will bind to the substance, and it will open only because of binding of that substance, allowing it to move through the channel. Tonicity, so you remember, a hypertonic solution is one that has a high solute concentration outside of the cell. So therefore, there's net movement of water out of the cell, causes the cell to shrink. If it's isotonic, our solute concentration is going to be the same. If it is a hypotonic solution, there is a higher solute concentration inside of the cell, which is going to cause water movement to go into the cell, and the cell will swell up. If left unchecked, the cell will burst. Same thing with hypertonic solution. If left unchecked, crenation will happen, the cell will shrivel. Now, the concept of osmolarity is tightly linked with the concept of tonicity. If something is a high osmolarity, that means that there's a high solute concentration. So usually water is going to move from areas of low osmolarity to areas of higher osmolarity. In the case of a hypertonic solution, that means that we have high osmolarity outside of the cell. So what's going to happen? Well, the water is going to move to the area of high osmolarity. So a high osmolarity solution is one that is hypertonic. If there's a high osmolarity inside of the cell, now we have net fluid movement or net water movement into the cell. So we have moved water across the cell membrane, and we have a high osmolarity here. So high osmolarity inside of the cell is a hypertonic solution. High osmolarity outside of the cell is a hypertonic solution. If the osmolarities are equal, then we have equal movement of water in and out of the cell. We are now an isotonic solution. So remember our tonicity model. We have three terms here. We have hypo meaning lower. We have iso meaning the same, and hyper meaning above. So hypotonic situation, we have the extracellular fluid that has a high concentration of water. In a hypertonic solution, we have an extracellular environment that has a high osmolarity. So we have an intracellular environment that's going to have a high concentration of solute. And in an isotonic solution, we have both the extracellular and intracellular space that have an equal amount of osmolarity. So therefore, there is no net movement of water into or out of the cell. There is simply going to be a one-to-one exchange of materials from inside and outside of the cell. And these are the main concepts of cellular transport. So if we remember, we had simple diffusion. We had diffusion that was facilitated by either a carrier or a channel. We had osmosis, which is simple diffusion of water. It can be either across the membrane as simple diffusion, or it can be facilitated by a channel known as an aquaporin. We have a primary transport mechanism, whether that be active primary transport, utilizing ATP to transport a primary substance across against a mechanical or chemical concentration gradient. And we have secondary active transport, where we utilize the energy that we cleaved off for the primary mechanism to allow for a secondary product to now move. And that movement, if it is going to be in the same direction, is known as a symporter. It's going to be in the opposite direction, is known as an antiporter. Our examples were the glucose-sodium link transporter. Glucose moves in, sodium moves in. The antiporter example, sodium moves in, hydrogen ions move out. Antiporter. All right. So that was a lecture covering membrane transport. Thank you all for listening, and I'll see you at our next lecture video.